I'm Faith.
Hi, I'm Elshaddai.
Hi, I'm Anchal. And we are members of Relay for Life. Relay for Life is the college chapter of American Cancer Society, and as part of the mission delivery team, we hope to spread awareness about cancer, not only by organizing fundraisers or by forming great relationships with cancer victims, but also by educating people about cancer research.
In my laboratory, we're very interested in what happens when cells are not actively dividing and making a new cell. So lots of cancer research focuses on cells that are dividing and making new cells, but we're really interested in what it means to be a not dividing cell at any time. And we think that this is really important for cancer research because a lot of different types of tumors show something that people call tumor dormancy. So you have an initial tumor, and then sometimes many years later, the patient will have a recurrence of the tumor. When I was a graduate student, I got very excited about cancer research. And when I was a graduate student, I was very interested in understanding if there were chemicals in the environment that were causing mutations that were giving rise to people's cancer. And so we spent a lot of time looking at the pattern of mutations caused by different chemicals in the environment and the pattern of mutations that are actually in people to try to see if we could find the most important mutagens and carcinogens for people. So that's what I got excited to study initially. So I think that some of the major breakthroughs in the last couple of years in cancer research. So first of all, we've had the ability to sequence tumors. And so with the rapidly increasing capacity for sequencing DNA. A lot of people have applied that to understanding what the mutations are in tumors, and this has been very exciting. Another big change that I think has happened in cancer in the last couple of years is the realization that cancer cells, they're not only dividing inappropriately, they're also growing. Because you can imagine if you just had a cell that kept dividing, it would keep dividing, you'd have lots of cells, but they'd be very small. So actually cancer cells not only divide, but they also get big. And actually that cancer is associated with a reprogramming of metabolism along with kind of inappropriate proliferation. I think the biggest challenge facing the field now is to manage all of this sequencing information. So I hear from patients and patients' families that even if their parents or their loved ones tumors have been sequenced, and even once we know the mutations, it's still not clear how to personalize the therapy. And so I think that's kind of the biggest kind of challenge for the field right now, is to go from that DNA sequence to a really effective cure for each patient. Oh, all the time. We have many setbacks. I mean, when you do research, every day there's all sorts of issues that we need to address. Today all the cells are contaminated. The next day there are Western blots that don't reproduce. Doing research is always about kind of dealing with challenges. If you're kind of at the cutting edge of anything, you're always having setbacks. But we try to deal with all of them in the same way, which is first of all to be calm, and the second of all to try to be data-driven. We develop models of how we think the cells are working, how we think cancer cells are working, how we think that other cells were working. And then as we generate more data, we try to think about whether it fits our model. And if our model doesn't work, then we try to come up with a new model. So our main goal is to try to use the data to help us to develop our models and to not be so in love with our models that we don't give them up. There's a lot known about kind of the cyclin-dependent kinases that are really important regulators of the cell cycle. And so we have a good understanding of that. What's less well understood is kind of what happens to cells when they exit the proliferative cell cycle. So that's part of the reason why we're really interested in that, is because we think that there are important things for cells to do when they're not cycling, but there just hasn't been as much attention given to that. Challenge is that we don't have great markers. So we don't have great markers for what it means to be a quiescent cell, and so we haven't been able to kind of pull the quiescent population out from people. And so one of the things that we're working on in this laboratory is finding better markers for the quiescent cells so that we can identify them and ask what's special about them, what's making them tick, and what's regulating them. One is things that target things to tumors. So a lot of the times we maybe have a great idea for how to treat the tumor, but it's really hard to get it to the tumor. And there is some very exciting work on kind of tumor targeting. Right now in my laboratory we have a medical doctor who's from Israel who came to do research with us, but she's actually treated cancer patients, and we brought her here in part because she would have kind of a clinical perspective to offer to us. Right. So I think, for myself, I think that there's a really important role to play in cancer research of basic biology. I think that, we still have the opportunity to learn really fundamental things about cancer in the laboratory, and that some of these will turn out to be translatable in ways that aren't really obvious from the outset. I also think that another field that I think is really exciting that's emerging is the field of immunotherapy. So there's been a lot of advances in using a patient's own immune system, kind of engineering their T cells to specifically attack the tumor and giving those T cells back to the patient.